Okay. Good. Yeah, it should work.
Looks like the video's working, right?
Yeah.
Okay.
Yeah.
Karna, did you start recording?
Yeah, it is
Oh,
going.
on YouTube, so the YouTube records it for us.
Okay. I guess maybe I can just just speak to Agnese about this. So the point of this is having as many students. So it's not like you invite just a bunch of faculty and then have this like very kind of thing. Like the point is to just have students get exposed to all sorts of different kind of theoretical work in biology and biophysics. So last week we had Yane talking about how is the length of actin cables and determined, and how do you have allometry and scaling with cell size? And today, obviously something very different. So the point is more just to expose people to lots of different things. And then once you've given your, talk, we'll shut down and have, we'll shut down the live stream and just have other kinds of questions, like science questions, but also like non-science questions.
Okay.
So think about it very much as a student seminar rather than like, and now grumpy, grouchy old people are going to,
Great.
You know what I mean?
Yeah, absolutely. And do people interact during the talk? Should I expect to be
If you'd
talking?
like that, then do you want that?
I would love that.
Okay. So definitely.
Okay, great. Because also I have definitely too much stuff, and I'm very excited about all of this. So I will put too much stuff on this. So please slow down as much as you can and as much as you want, because otherwise it will be a lot. But yeah, I'll try to do my best to not be too excited and try to be clear.
And I would just ask everyone that can and is happy to share video to try to do that, just because it does feel a little bit.
It helps, yeah. It helps.
A bit more human, which is something we should all try to cultivate. So if you're happy to do that, please do so.
And also, if you would like to ask a question, which we encourage you to do during Agnese's talk, like you could unmute, or you could raise your hand, and then we could stop her at an opportune moment, and then you can unmute and ask the question.
Yeah.
Okay, I don't have to take care of that, right? You'll see.
We'll take care of it.
We'll deal with questions.
Yeah.
Okay.
Don't worry. Should we get going? I could introduce you. If that works. Okay, cool. So Agnese started, I hope I'm pronouncing that okay, started out in science, studying like the really kind of scary stuff in physics, like, I don't know, at least to me, like fluid dynamics and turbulence, these really difficult differential equation stuff. And as it applied to like weather and wind and cloud formation at NISA and under Antonio Solani and Andrea Mazzino. She got her PhD in 2007, and she kind of switched topics and locations a little bit, moved to Harvard to work with Michael Brenner, and started thinking about things like biology, thankfully for us, like the relationships between biofilms and complex fluids, how fungi get around. She also did a fellowship with Massimo Bergasola and was an instructor at Harvard. She started a lab, I believe, at Institut de Physique in Nice in 2013, but is now at University of Genova in Italy as a professor in fluid dynamics, examining like the importance of fluid dynamics throughout biological systems. So we're really excited to hear her talk about olfactory navigation today. You can go ahead and correct me if I said anything completely wrong.
No, it was great. Thank you. Thank you very much for the invitation. I'm sorry this is online because I like to give talks in person, but I'll try to make it as lively as possible. You will see my hands a lot. I'll try not to wave them too much, but I'm Italian, and so I can't really cope without my hands. I'll talk about olfactory predictions and navigations. It's something that I've been interested in for a while. And I think right now there is a combination of things that make it very exciting for me at the moment. There are a lot of experiments going on, and I know this is a theory seminar, but I can't really do theory without data a lot, especially in biology, and so I'll show you. Sorry?
I think someone.
There isn't anymore. So I would give like the phone to Marcus.
I think maybe everyone should mute their own stream so that we can hear Agnese. Okay, sorry.
Oh,
Agnese, go ahead.
it was not a question. Okay. I was surprised. It's a little early to ask questions, but I'm happy to answer any questions at any time. So please do interrupt. Okay, so as I said, I'll give you data too, because I think it's the interesting thing about biology to me a lot is the fact that there's so much unknown that you really need to go back and forth between theory and experiments a lot. And this is it's really exciting. It's not a field where you have the questions that you have to answer, like this big list of big problems. You just have to interact a lot with the systems to ask questions. So I'll show you experiments and theory across different systems. It's not a point where I can give you a talk where I ask a question and I just develop it through the talk and give you the answer, but hopefully it provides enough ideas that it interests you in the topic. I don't have definite answers, more questions than answers. Okay, so the general topic merges fluid dynamics and biology in the following sense. We are all systems that live in a real world, and so we are immersed in fluids, and fluids give us information, and we use this information for a lot of things, including navigation. We need to reach food, reach mates, escape predators. All organisms do this in different conditions. Large organisms like us do this in a typically turbulent environment, and so there's a lot of noise. And so the reason why I think navigation in turbulent environments is interesting is because it's a paradigm of what do we do with noisy information, right? And there's a lot of examples where we have now tools to navigate noisy information like Google search. What would we do without Google search with all of the information that's on the internet? It's just very hard to find what's useful to you. And just turbulence is a little bit maybe more treatable than Google search and the information on the internet, but it's the same kind of idea. There's noise, there's information, there's a goal. How do you reach it? Okay, and to move forward and beyond this very qualitative ideas, here's the plan for today. I'll show you fish, I'll show you mice, and I'll show you octopuses. And this will take us through three main ideas. One is the idea of multimodal navigation. The other one is try to quantify how hard is navigating turbulence. And the third one is making predictions. Okay, does that not seem to work very well? Okay, number one, fish. Okay, so I've come across this interesting fish, and I realized after I wrote a long project about it that this is something I eat all the time. It makes a really good soup. It's called gallinella in Italian. In English, it's called sea robin. So here's the animal. As you can see, it's a normal fish, but it has something special that's a little creepy to see. It has legs. So it not only swims, but also walks on the seafloor. And here you see it a little bit better. These are experiments that my collaborator are doing at Harvard. It's a big tank, and the fish is moving. And here you see it as it walks. It also swims normally as other fishes, but it also walks. And the reason why it walks is not just to slow down or get rest. It's because, here, let me show you the more magnified version. On the left, you've seen the zoom out, and it's sped up. On the right, what you're seeing is the real speed, the normal speed. And you can see, maybe I'll show it again. You can see that it walks because what it does is it digs and it finds interesting things underneath the sand. Now, this is an interesting property because there's a lot of stuff underneath sand, and apparently this fish is so good that other fishes will follow them to get their prey because they are very voracious, they're very good at this. Okay, and so they developed these novel sensory organs, these legs, to find prey buried underneath sand. So how are those legs? What is their structure? So my collaborators are physiologists, basically, and so they're interested into what the cells are that make this tissue, how are the nerves connected, and what is the current through the nerves, depending on what the signal is. So they're really interested into the details of how this functions as a sensory system. So they've done a lot of things, and one of the first things we've decided to do is to check their sensitivity to chemicals, because it's the first thing you think about. And it turns out that these legs are chemically sensitive. And basically, right now we're thinking about this as taste. It's the structure of these legs is kind of like similar to taste buds, although they're not really taste buds. There's a lot of details that we're trying to understand right now. But basically, through this taste or odor or chemical receptors, they're able to find this prey buried. Here, what you see is on the left is the sketch of the experiment. It's a reasonably sized tank. It's 1.5 meters in diameter. And you have sand underneath, you have water on top. The fish can do whatever it wants. And then at some point, we dig a prey underneath sand and we look whether they find it. And it turns out they find it, even if it's buried pretty deep down, not at 5 centimeters, but even 3 centimeters, they find it. And so now these are experiments done with a mussel. So the mussel is a complex prey, and it has a lot of chemicals in it. And so it's not really a very well-controlled experiment from the point of view of the odorant. And so what we did is, and when I say we, I say they, my collaborators found what the chemical is that they respond to. And it's called betaine. It's present in a lot of organisms in the water, and so it makes a lot of sense. We checked that they respond to betaine both through behavior by making these capsules full of betaine, concentrated betaine, and they find the capsule even without the mussel. So behaviorally, they use it, they use this chemical. And then what you see on the bottom is this dose response curve. So you can flush some concentration of this betaine directly to the leg, and you can measure the current through the nerves. So you can see if the leg responds to this chemical, and it does. So the question is this enough? Is this enough to find the prey buried in sand? Is this kind of like taste sensation enough? And so what I did was I made it the simplest possible assumptions. I made a Poisson searcher model where what the animal does is it swims in the water, and then it randomly checks on sand if there's something interesting with a concentrate. And if it touches sand right above the capsule right above the target, it will sense something. It will sense some chemical signal. And at that point, it will dig and find the prey. So the rate of success of this Poisson system depends on the area on sand where the fish can sense the signal. So basically, everything in this model can be computed. There's very little uncertainty. So here, it's basically a first passage time. Since this area changes in time, it's not homogeneous and it's continuous time. So we can compute everything from the probability density of the first passage time. The first passage time means the first time it hits the area where it senses the odor, right? So everything is basically known for this stochastic process. And what we have to compute is this lambda of t. Lambda of t, you can see my arrow, right? If I move it here.
Yes.
Okay. And what is lambda of t? Lambda of t is the basic rate at which the fish switches from swimming to touching the sand. This is a number that we'll assume constant. And then it has the ratio of the surface where a fish can sense something. It's this kind of like a green cyan arrows here. It's S of t. This changes in time because there is this fusion. And then on the denominator is just the total surface. And then we have a threshold. So this, to define this S of t, this area where the fish can sense something, we'll have to define a threshold. And we have this from physiological data, okay? So-
Agnese, if I could pause for a second, we've got a couple of questions already.
Yes.
Ananda, if you wanna unmute and ask your question.
Hi, just in your previous slide, so there were two plots. Yes. So how do I understand this kind of very sigmoid kind of response, and what is behind, kind of like why there is this huge concentration dependence to a certain level?
Well, so the way we are thinking about it is this is the threshold. It doesn't measure anything below a certain threshold because any biological system will have a finite threshold, right? So on the left side, this is basically noise. And that's why I'm showing the blue as well. So this is just current that goes back and forth because of noise, because of something else that's not related to the signal. Okay, and the reason why there's saturation, there's a lot of times there's saturation. It depends on what the system is, but this is a typical, it occurs all the time in olfaction as well. So for olfactory receptors. And so basically the only thing, I'm not really thinking about the curve too much, the details of the curve too much, but I only want to point out that there is definitely a threshold below a certain value of the concentration. There's just no response. So I mean, unaware.
Yeah, the reason I'm asking because it seems like very similar. If we have a transcription factor, it finds a target sequence on a DNA, and so we do also see similar behavior with the concentration response. So that's why I was
I mean, sigmoids are really typical. If you think about it, this means simply that a concentration that is beyond a certain value, the response is constant, which is very reasonable whenever you have a finite number of binding sites. You have a finite number of molecules, whatever the response it is that you're measuring. Here it's current, so there will be ions that move back and forth. So possibly there is a finite number of ions that can be employed for this system. So it's not, it's very general. I agree with you, but I don't have particularly deep things to say about this. It's widespread in biology. Sigmoids are there. Yeah.
Okay, and Srijit?
Yeah, so I wanted to ask if there's any secondary smelling organ in the fish. You mentioned that the leg senses the smell. Is there some other organ within, like, let's say a gill or like fluid flowing through it that senses the smell?
Yeah. So it's a normal fish. So it has a nose. It has all of the apparatus, the normal smelling apparatus of all fish. It has these organs. These are extra organs, extra chemically sensitive organs. And it's precisely a question, why do you need this to be chemically sensitive, and what's the interaction with the nose and the more ordinary olfactory system that is present in this fish as well. So it will come in a second, but I don't exactly know yet how they interact. The bottom line from here is that I think they do interact.
Okay, yeah, thanks.
Okay, but it was a great question. Yeah, it's basically what I'd like to know too. Okay, so the null model is a Poisson searcher, and we have these 3 parameters that we need to estimate. The first, so to summarize, lambda 0 is the rate at which it touches the surface. It transitions from swimming to walking. S of t is the surface on sand where it can sense something, and this depends on the threshold, on the sensitivity of the legs. So the rate will I just analyze trajectories from these animals. This is a little cumbersome, and we'll make it better because it's not automatic yet. So the analysis is kind of painful. You have to go through the movies and look when it's walking versus when it's swimming. And this is an example on the left. We have a bunch of trajectories, not infinite, also because the analysis is not very easy to do. So we'll have to automate it to make it better. But on the left, you have an example of this, of a trajectory. And in purple, you see when it's walking. And so you can compute the transition, the rate of the transition from swimming to walking. It's not a beautiful exponential, but it's kind of exponential. And so I can get a rate from here. It's every 15 seconds. And I can also look how long they stay on sand to make the model a little bit more precise to calculate this, the probability that it touches something. But I won't go into these details because it's just, there's nothing particularly interesting about it. And so the second parameter is the surface, where the fish can sense something. And this is just modeled as a diffusion process. It's not perfect because this is sand, and there will be potentially some flows, like the flows that flow through porous media. I have neglected this, but it's potentially important. And by doing this, you can compute everything. This is just a diffusion process, and also the boundaries are far enough that basically this is just the solution in an infinite volume, basically. And so you really can compute everything. The detection threshold comes from those response curves that I showed you already before. And the result is basically niet. There's no way that this is a Poisson process. What you're looking at here is the predictions of for the Poisson searcher for the probability to capture prey, depending on what's the concentration in the capsule. And on the right, you see the time, the average time it takes to reach the prey, to dig for the prey. Okay, constrained on the episodes that were successful. And what you see is a comparison of the lines, that's the theory. And I spent 4 orders of magnitude in terms of thresholds to be like extra sure that this doesn't matches. But there is no way that I can match the theory and the experiments. The experiments are the stars. We have, as you can see, we have basically in terms of successful trials, we have 7. So it's not a lot, but it's not 0. And we tried for each concentration, we have 10 trials. Some of them are successful and some of them are not successful. So basically the answer from here is, I know what they're not. They're not Poisson searcher. And there's many ideas that we're exploring right now. The simplest idea is what somebody before asked already, which is what about using your nose as well? Because this odor gets out from the prey, it diffuses through the sand, but then it gets into water, right? And once it gets into water, then you can use your nose, which may be more sensitive. Maybe you can get the signal from a lot of different places. And so what we did was we tested whether behaviorally we saw a difference when the tank, there was nothing basically in the tank, but we would add some mussel juice in the tank, in the water, okay? So in the bulk. And what we saw is that the fish would be sort of like alert to this, and it would go back and touch the floor more often. It would actually not more often, but more often, sorry. It would touch the floor more often, which means that there is some signal that comes from when it swims. It's not purely random, it's not purely Poissonian. It combines the nose and the legs, okay? This is the simplest assumption. It might not be true. There are some other ways that this could work. For example, as the fish moves through the sand, like close to the sand, it will pump basically water from the sand. And this will increase the amount of odor that comes out of the sand. So this could be something that also plays a role and potentially changes the predictions. But because of these experiments, we now think that it's really an interaction between the two. Okay, and so as I said, this is the beginning for me of this of these experiments of this system. So I don't really have an answer, but I do think that sea robins use a multimodal strategy to get to, even to dig prey. They definitely must use it to find prey in the water column as well. They can use mechanical sensing and chemical sensing. They have a lot of mechanical sensors along the surface of their body to measure what's the velocity in the water where they swim through, velocity and acceleration. So there's a lot, it's a very rich system that I'm just starting to explore right now. So there's not a lot I can tell you about, but I definitely think it's a combination of different sensory systems. And we're working on this. Something is coming out soon, but we have to work out a little bit more things. So I decided not to tell you more about this. Okay, so so this is an interesting system that one of many that uses chemical cues in water, in a fluid, to do navigation. And this is, in the real world, this is hard because of turbulence. As I said in the beginning, turbulence makes this kind of signals very noisy and especially very sparse. So this is, I'm gonna tell you something about how hard this is through another system that we worked on a few years ago now.
Sorry, Agnese, before you move on, can we stop for, we had another question from Nan, actually.
Yeah, thank you. So I was wondering about this detail about multimodality, right? So you need probably, you can think of having two or multiple sensors. They take inputs, and depending upon the combination of inputs, now they will decide whether they should go for that food or they should run to save their lives. So how much we could understand from this perspective about sensing?
Wait, so you are talking now about the identity of the odor?
Yes.
Okay, odor identity is something that has different constraints. It is it's a different goal, right? And so one struggle that I think people have in the literature is that if you look at neural data, if you're interested into how the brain works, you can find different results based on what the animal is actually trying to do. What you suggested is you have to classify the odor, the source of the odor. You have to classify to understand what is there, a friend or an enemy, for example. And that problem has a different set of constraints. But so on the one hand, computationally, there will be different constraints. And there's a set of really interesting work about how do you measure the presence of a mixture. Whenever you have a complex, a real source of odor, a real target, it will emit a combination of odorant, of single molecules, right? And so you might want to infer what the identity of the target is based on what is the composition of this mixture. And so there's a set of interesting problems about how do you recognize where the source of a mixture is and what is the composition of the mixture. This is based on how different odorants will be transported by the same flow. So it's based on correlations. And there are some recent results that really suggest strongly that the brain is able to measure correlations. So to go back to your question, it looks like part of the hardwire of the wiring of the brain is made to answer your question, is made to know where do mixtures come from, and mixtures potentially identify targets. So it's the label that you give to your target. That mixture is a friend, this other mixture is an enemy. So in the real world, this is a complex problem because enemies and friends are not single molecules, but they're mixtures. And I think that the animals will care about this task. It's a different task with respect to what I'm gonna tell you about, but it's a very interesting task as well.
Okay.
I don't know if this helps a little bit. Okay.
Sorry, we have one more question from Joseph. Yeah.
Hi. Yeah. My question's about you sort of rolled out the Poisson-Walker model. Are there other models that only use one, not multimodal, only use one mode, just taste, for example, that could possibly explain the data?
Well, if you don't have any distant cue, if you only have touch, so this is the dichotomy, right? There are senses that are distal, and there are senses that are not distals that require you to be there. If you don't have any distal sense, I don't know how could this be different from what it is. You could have memory, for example, about where you are, where you've been, but I've input this, I've modified the model to account for this with excluded volume, and it doesn't make a difference. Actually, what I showed you in the image, that's in the results, that's the result of taking into account everything I could possibly take into account. The surface of the fingers, I can't think of anything else, because if you don't have any distal sense, there's no homing. You need to be to get there.
You need to get there.
Right. Apart from a memory of where you've been, but it doesn't restrict enough the possibilities. So I really can't think of anything else. If you have ideas, please shoot. Are you thinking of something specifically?
Not really. Yeah, it's just like I just was curious, like, you know, how many different models you chose. But like, yeah, I mean, you know, I mean, the legs are also going through the water presumably at some point. So can the legs, you know, the legs are also going through the water.
Yeah.
Right. So can the legs, you know, can the legs sense the signal just in the water and not through the sand?
Yeah, absolutely. So
okay.
they could be the ones who also who measure both in water and absolutely. When I say multimodal, is because the signal in water, it's about the signal, not too much about the sensory.
Not the organ. Yeah.
I think the nose will be much more sensitive than the legs. That's what my physiology friends tell me, and I think so. I think if it's to sense something in the bulk in water, I think they'll use the nose, probably. But it's about the signal itself. It's about using something that's in water versus something on sand.
Got it.
Okay.
Yeah. Thank you.
Are there any other questions?
Yeah, let's take one more from Manuel.
Thank you. My question is kind of in the opposite direction of the previous one. I wanted to know if there's like a known example where the Poissonian model works, like some simpler paramecium or something like that actually functions as this Poisson searcher?
Well, so in, for example, chemotaxis is basically a Poisson process where you have modulation that's based on your past history. But it's a short past history. So it's basically, you can think about it as a Poisson searcher. It's at small scales, and it works. Basically, so maybe I should make a step back and say, I did not necessarily think this would be accurate, but you need to have a null model before you can say that something else is happening.
Sure.
And so the reason why you put out a null model is to start to think about a problem, right? It's a null model in the sense that it's the first thing you try to start and eliminate simple scenarios. If it was just a leg, I don't know how else it could be. If it's just touching, it needs to be, and there is no distal sense. I don't know how else it can be apart from responding locally to the signal. So it can have memory, like a complicated way of having memory. I included it in a sort of like cheating a little bit because I still use the Poisson model, but in the rate, I included the fact that there was some excluded volume coming from the fact that the fish had already touched some part of the tank. So it's not really memory in the sense of non-Poissonian. It's still Poissonian, but with modulation in the rates. Yeah, I really think there's something more to this, just because I can't think of something else. But please, I welcome ideas, and if you have ideas to make this simple model more accurate, then I'll be very interested to know.
All right, thank you.
Okay, this was part 1 of 3, and I think I'm pretty much done with time. Is that right?
No, but I think what we want to do definitely, I think by 1:30 is have the sort of more broad discussion about, and so maybe
by 1:30, so another 20 minutes.
20 minutes.
Good.
So
So
yeah.
So let me move to this. I'll move quickly through this intermediate step, which is how hard turbulent navigation is, mainly because maybe people don't know about this. And so although it's very introductory in this context, maybe it's useful, but please tell me if this is too basic or too well-known. Okay, so we've mentioned chemotaxis before, and this is just an attempt to make it a long story short. See here, so the question is how hard it is to navigate turbulence. And so the basic, the reason why navigating turbulence might be hard is because C, the concentration of an odor, is transported by V, the fluid flow, the velocity of water in this case, or air, or any fluid, and it creates complex structures. This depends on this parameter P, Peclet, which is a number that tells you whether advection or diffusion dominates in the system. Okay, so this is a very well-known equation. It's called the advection diffusion equation, or passive scalar transport, depending on where you come from. What else should I tell you? F is the source or sink of odors. In our case, there's always a concentrated source. Okay, and if this Peclet number is small, then that means that advection is subdominant. You can forget about this complicated term, which is linear, but it's convective. So there's this velocity that makes everything complicated. So you can forget about this. And what you end up with is an odor that's smooth. And then the smooth odor has the nice property that the gradient points to the source. And this is exploited over and over and over by a lot of different organisms that live at small scales, typically. And that's because if you look at it, L is here. If you are at a small, if you care about small length scales, you typically are at small Peclet, so you can hope to do just gradient climbing. And something else happens in the opposite limit where Peclet is much larger than 1. So you can see it's large velocities, large scales, small diffusivities. And this is where this gray term makes a mess, okay? So this is the simulation that we are doing right now. We're working on this field. And what you see here on the bottom in white, there's no units because I don't really care about the units. That's how it looks like if you stand in between this field at where there is the white point, and you just stand there and measure the concentration of the odor. That's what it looks like. It looks pointy, goes up and down all the time. This property, which is a very fundamental property of turbulence, is what makes this very hard. Despite this, there's a lot of animals that use information from turbulent odor cues. And so the broad question is, how do you extract information from this mess? What is important to measure? And how do you clean this from noise? Okay, so I wanted to elaborate a little bit on this, and I'll show you just a quick experiment to tell you a little bit more. This is something we've done a few years ago. Here you see a mouse, and this is our arena. It's pretty small, it's 50 centimeters in size. And we have, in the green dots, there is a source of an odor, and we teach the animals to find the odor. And when to teach it, the animal learns that if it finds the odor, it finds water. And because it's thirsty, it's motivated, and it does it. Okay, and so we found a lot of interesting things about the behavior of these animals as the task would progress. But I'll just limit myself to a very simple observation, which is that, so here what you see on the right is the progression of their performance. They get better and better and better. Here on the right, when they're late in training, that's where we found a lot of interesting behavioral components to this task that I'm not going to tell you anything about. But early in training, that's where I started. And again, this is a non-model where I just wanted to show that gradients would not be enough to get to the source of the odor. And so I did the simulations that you see on the left here. And then I just coded a very basic stochastic gradient climbing algorithm. And I realized that it finds the source, which was unexpected coming from the assumption that turbulence is hard. But in retrospect, it's understandable because this is an in-between problem where the Piclet number is quite large, although not huge, but more importantly, we are at small enough scales. At these scales, what happens is that if you look at the direction where the gradient points, it doesn't always point to the source as it would do in a smooth gradient, but it often points to the source or in the right direction. And so basically you perform a biased random walk that gets you to the source. Okay, great. Is this the end of the story? Well, the lesson from this that I want to draw from these results is that this is only possible at very small scales. And the reason is because as you move away, the signal becomes more and more intermittent. So basically you don't have it all the time. And so the gradient just loses information. The noise becomes too dominant, but it's not noise itself, it's sparsity, which influences noise, of course. So sparsity is what makes turbulent navigation hard. This is the message I just wanted to sort of go through in this intermediate part. I have another point about this, and it's that laboratory experiments typically have arenas. They're not made in the lab. You don't let animals run into the lab at random. You typically make arenas. And so arenas are typically small in size. And so one point that I'm trying to suggest to my collaborators is that if we move at larger distances, we'll find something interesting about behavior. There's a lot more interesting things happening at large distances because these basic algorithms don't work and the problem becomes harder. And this is something we are working on right now, but I decided not to tell you about it because it's already too long. So, okay, so turbulence is hard because of sparsity, but on the other hand, sparsity, if you think about it, can bear some useful information. So is there anything that we can grasp from measuring sparsity instead of just being drawn back from by sparsity? Let's just embrace it. Okay.
Before we move on, I think Salam has a question.
Yes.
Yeah, I was just wondering, does the fact that the spatial resolution by which you can sense the smell, does that also complicate things when there's turbulence because you will be averaging over a bigger area?
Yeah, so typically the time scale that they measure, that they sniff, that mice sniff, is on the time scale slightly faster or of the time scale of the Kolmogorov time scale. So that means that typically you are, so first of all, averaging doesn't really change much because noise is very small, very slow to average out in turbulence because you have these power laws. So it takes forever to average out. And I have some results on this, but I can show you later. In terms of the volume that's inhaled, it's not much larger than the volume, the smallest volume in the turbulent cascade. So that means also that it can be a little larger, but not much. So that also means that there is some averaging, but it won't change dramatically the picture. Yeah, maybe if you give me a moment, I'll show you in the next section what's the effect of memory. So how well it converges if you increase memory.
We have one more question from Vasil.
Hi. Yeah, very interesting talk, first of all. And question. So you mentioned that this turbulence sensing is much more difficult for obvious reason. And the natural question that arises is time difference between peaks actually a quantity that is being measured by an animal? Because naively, like if you're trying to imagine like how would you find something that has that like turbulent odor pattern, obviously, like you go somewhere and you sniff, and then you go to another place and you sniff, and if time difference between peaks is larger, presumably you're coming closer and closer. So,
absolutely. Let me keep going because there will be an answer to not really an answer, but we'll talk about this. Yeah, it's a great question. So, okay. So let me show you just one last movie because this is what inspired us to ask a simpler question. So we'll move away from navigation and we'll talk about predictions. That's a simpler problem in the sense that it's static. So what we'll ask is, assuming that I'm a point, I have one sensor and I have a certain memory, can I infer what is the source, where is the source of this chemical substance in a turbulent field? Okay, and this question was inspired by this phenomenon that's been observed already. These are experiments that my collaborators at UW do, also in this great laboratories at in the islands, San Juan Islands. So I very much enjoy going to visit. So this is an octopus. And what you see, so octopuses have been described to do this behavior where they stay still, potentially accumulate information. I don't know if you know anything about octopuses, but they are fascinating because they are like aliens. They have these 8 arms, they collect information from the arms. The central brain is very small, so there is a doubt if it can handle all of the decisions, and potentially these are organisms that have a distributed system of decisions. They have a vague perception of where their body is, as opposed to us. We have a perfect perception of where our body is. There is a number of interesting things that we're hopefully gonna get to, but this is not about it. This is just an inspiration and comes from the observation that octopuses often wait, accumulate information, and will just go directly to catch the prey, basically in a straight line. Now, this is a large octopus in a small aquarium, but it's been described in the field as well. So the idea is that octopuses might be able to predict where the prey is without approaching it. So from standing from one location. Okay, so now in the next 5 minutes, I'll try and tell you something meaningful about what we've done next. So this is the premises. So the question we ask is a specific question. If you are there and you measure the odor in time for a bit, what should you measure? What is the best feature that you should measure? And on the bottom, you have exactly the measure that, what was his name? Somebody just asked about. That is the neuroactivity. It's a demonstration that there are some neurons that keep track about the timing of odor encounters. When was the last time you encountered the odor? Okay, this is in lobsters, it's called burst olfactory neurons. They measure timing. And this is an example, but it's more and more clear that the brain can measure time. And on the top, this, let me skip the details, but it's just a measure that indicates that other neurons in the brain, especially in insects, but in many other systems, can have activities that's proportional to the concentration of the odor. So are measures of intensity or timing more useful for making predictions? This is the question. Okay, and to answer this question, we made this large-scale numerical simulations. We have those snapshots in space and time of the odor. We segmented in many segments in time series that can look like the one on the left or the one on the right. There's a mixture, very sparse, less sparse, noisy, less noisy, more intense, less intense. It's a realistic combination of what can be exposed if you are trying to measure odor in space and in this situation. And then we take 5 scalar measures that will quantify this time series. So we have a certain memory, things change with memory, but not a lot. And we have 5 measures that are described right here. The first 2 are measures of intensity, so average concentration across your memory, and then average peak slope, that's the slope and when the odor first comes on. So here you have, I don't know, 4 or 5 whiffs, and you measure the average. These are 2 measures of intensity, and then we have 3 measures of timing. The average duration of the whiff, so of a time where the odor is detectable above threshold. The average duration of blanks, so the time where you measure nothing. And the intermittency factor that we've seen before, it's the total fraction of time that the signal is on. Of course, the average of these last 3 features is connected, but noise is a little more complicated. Okay, and so what we've done is we did supervised learning. So we took a lot of data from these simulations. We taught an algorithm to learn from examples of input and output. The input will be these 5 features, one at a time or in combination. And the output will be the distance of the point where you're measuring the odor to the source, okay? And we can do this in the two dimensions, span-wise or downwind. And then here is on the bottom left, let me skip this. It's, we identify the minimal ingredients for this algorithm that will allow us to rank these features, to understand which one is better. Okay. This is the way we quantify the error. So basically here on the right, I'm listing the various steps of this algorithm. And once you have trained your algorithm, you train it on a new test set, on a set of data that the algorithm hasn't seen. And you quantify the predictive power of that feature by measuring the mean square error, and we normalize it. Okay, and here on the right, you see a way to quantify this. It's plotting the actual distance, so the real answer, and the predicted distance, so the f of x, the answer that my algorithm spits out. And here, the light gray is an example of a situation of one of the situations we tested. So these are all of the points that the test points are in gray. If they were all aligned on the bisector, this would be a perfect prediction because predicted equal actual distance. In this situation, the error is 0. When the error is 1, then it means that you have a horizontal line, your predicted distance is constant, and it's the average. So it's not like you learn nothing, but you just learn what's the average, the typical average of the distance. So it's a trivial prediction, okay? So it can even be worse than 1. The error can be 2 if it's a random, completely random prediction. You don't even learn the average, you just learn the boundaries. It can be even worse. If you don't know what the boundaries are, you might predict that you are 2 kilometers away. The error is huge, okay? Okay, so let me go very quick about this because I just want to get to the conclusion. The basic result is that, and actually, let me remain here. The main result is that the best feature depends on where you are. There's not one best feature across all space and time. You need to know, you need to do something different as a function of what's the order that comes in. And specifically, so let's just look at this result. It will be true in X and Z as well. In this case, what we've done is we've sectioned our field. The order field is 3-dimensional, right? You have the source, you have turbulence, and in X, Y, and Z, you have this order field. We sectioned it in Z and asked the 2-dimensional question at different Z's, okay? And at different Z's, what happens is at low Z, the intermittency is, the signal becomes more continuous because you're getting closer to the boundary, which acts as a mixer, okay? For very fundamental reasons, right? And the signal is more intense and more continuous. In this situation, which is a simulation A, we are here, and you see that the light gray features, which are the intensity features, perform much better than the timing features. But as you move away from the surface, the signal becomes more sparse and less intense. And basically what you end up with is a situation where intensity features don't do very bad, but considerably worse than timing features. Okay, and this is a very important lesson that we drew from this work, because it means that an animal, it needs to either adapt to a specific niche. This is it's for a single flow, right? We're not even talking about changing the flow. In a given flow, depending on where you are, the features that bear most information about the source vary. So if you are an animal, you need to know this, and you need to either focus on the range that you're interested into, or be able to switch, or be able to measure both, because that's the other lessons that we drew from here. If you look at the green here, the light green are combinations of features from both classes, from the two classes. Okay, so what we found out is that basically the two classes are, let me skip this. The two classes are, within each class, features are redundant, but across the two classes, features are complementary. And so we found two classes of features that there's a lot of interest to combine, because if you combine them, you are putting together complementary information. And so one of the questions we have is, are there other classes? This was inspired by data, by neural data, right? But are there other classes that we can invent and that will have a complementary predictability on the source location? Okay, okay. Let me conclude with a couple of remarks. This is what I was rushing to finish to tell you, but I will not, because obviously there's no time. But we're thinking about how all of this generalizes to other flows. And this, I wanted to tell you this also because there is this very powerful formalism. It's called the Lagrangian formalism. And if you're ever interested into fluid dynamics and how an odor or a chemical in general gets transported from a concentrated source, this is a very powerful formalism that you should know about. And there are recent results about how this applies to the situations that, for example, insects are mostly interested into. And by using these powerful asymptotic results, you can demonstrate that similar behavior will apply to different flows, to different situations, different niches. So this starts to become something that biology can really care about if it's general enough. Okay, I will conclude because I'm above time, and let me just say that all of this, we're combining the different parts that I told you about. I told you about prediction, and then I told you about navigation. And the features that are most predictive are not necessarily the ones that are most useful to navigation. So this is what we're trying to do right now, using a multimodal algorithm for navigation to probe whether A, redundancy helps, especially in more noisy conditions where you have to correct for errors, and B, do the most predictive features of a turbulent queue help in navigation as well. Okay, sorry, yes, I went over time, but thank you very much, and please feel free to ask questions or to start the discussion.
Thank you, that was really wonderful. I think Manuel has a question.
Yeah, I don't know if my question makes sense, but I was just probably I'm projecting too much of my own experience into what these animals do. But I was thinking about sort of like the time scale of perception, of conscious perception, because when I smell something at a distance, like I just have this subtle smell. I don't see it like coming, oh, it's there, it's not there, it's there, it's not there. Like it might be turbulent, but what I'm experiencing is just a subtle smell coming to me continuously. So I wonder how that plays into this animal. Do they can actually measure the sparsity of the signal, which I assume has to do with how strong the molecules that they're smelling are binding to their receptors, and however many layers of complexity. But I couldn't stop thinking about that.
So I have two comments. The first comment is that you might have a conscious perception of what you're doing, and your brain might be doing something else, right? Your brain might be doing something more fine that you are actually aware of. There's plenty of examples for this. One that I really like is athletes, when they learn how to, for example, swing a golf, or different other motions, kind of motion control that are very fine and very precise and repeated. If you measure how precise they are, the precision goes far, it's much faster, it's much more precise than a single firing neuron. And so obviously this cannot be conscious, but your brain does it, right? So there's plenty of things like this. So you shouldn't think about your perception as an indication of what your brain is able to do. The second comment is that there are some experiments. So humans are not usually thought to be that good with olfaction, but actually there are some experiments that show that, for example, if you take something and make people blind and you ask them to go back to the same location based just on olfaction, the performance is not great, but it's considerably above chance. So that means you are in a room and you don't see anything, but there are, I think it was 4 different odors would come from different sides. And then your task would be experience that one location, then be moved away, and then go back to the same location. And I think the performance is considerably above chance, which means that we're not that bad, probably. And the last comment is that this is an experimental result. So in lobsters, they have demonstrated that there are neurons that fire depending on when the last whiff was perceived, the last time an odor was perceived. So this it might not be used at all, but it's an indication that the brain, at least the brain of lobsters, is able to quantify intermittency timing very precisely. This is not the only result. It's the one that I think is more clear, and it actually measures one of the features that we've looked at, like exactly one of those features. But there's many results now that show that intermittency is used by insects, for example, by walking flies. There are results now showing that they do use the tempo, the timing.
All right, thank you.
Yeah, sure.
That was great. Oh, we have one more question. Madhav.
Thank you so much. I have a not very precise question, with probably I'm not even sure I'll be able to ask it well, but there's this, it feels like there's this assumption, implicit or not, in this thing where the goal is to find the source, right? The goal is to, but I guess I'm asking, are there, maybe microbial systems or other systems in which, I can imagine, I can just make up some just so story that actually, the cost of motion is some metabolic cost, blah, blah, blah. I can make up some mumbo jumbo about how, maybe the thing that's being optimized is not necessarily get to the source. It could be figure out whether the source has enough calories as quickly as possible. I mean, you can imagine the function is something far more complicated than get to the source. I guess the question to you is, the empirical question is, can you imagine some, microbial, I only say because it's more experimentally tractable, some system where you can get a handle on what actually might be the sort of, the function, right? The thing that the system is trying to do, just because I can make up 5 or 6 metabolic stories and things, and I'm not sure which one is correct. It's not obvious that getting to the source is the single thing.
So, okay, I have a couple of comments. So the first comment is that you are perfectly right. So these problems extract and abstract a very specific question, but behavior is complicated because animals do what they want to do, and it's unclear. They don't tell you what they're trying to do. So, and it's one of the complicated things about neuroscience, at least the combination of behavioral and sensory neuroscience, because they might want to sense something because of reasons that you haven't considered. And so once you see the neural representation of a signal, you have to ask yourself, what do you think the animal was trying to do? And so this is just to say that it's a very good question. I have, so there's these experiments that I find really illuminating about ants. So ants, the ants that go fetch food to bring it back to the colony, those ants have a very specific problem, which is they have to have enough energy to bring the food back, right? So they have to make predictions about how much food do I have? How long do I think it will take me to go back to the colony? And how much should I eat so that I bring back as much as possible without risking to die? So this problem, there are very interesting experimental data where what they did is they followed the amount of nutrient within the belly by fluorescent methods in a real colony of ants performing the task that they were performing. And they monitored this in time to see what was the decision process behind the decision about how much to eat, right? You must make a lot of predictions. And this is where I think, so I really think that predictions and navigations are deeply intertwined. I don't think it's just a computational idea. There's this a lot of people will just analyze, I mean, a lot, no, it's not a large field, but I've seen people analyzing the amount of information that different features of an odor field will bear. And that's really not about navigation. Predictions and navigations are two different things, but organisms do both. And typically both are interesting, and the cost of making prediction and the cost of moving are different. So in the end, you really need to focus on the task in order to be able to predict what the decision should be behind this. I think I don't have more to say. It's just to say that actually there's another example. The other example is moths, because moths, which is a very famous example, in fact, because moths, so they will live a lot more in the larval state than in the adult animal state, and they don't eat. So there again, the goal of the male is just to find the female as fast as possible before dying. And that's I think that's a very clear goal. So minimizing time to reach target is very plausibly their main goal throughout this time. So whatever you observe in those animals will probably be dictated by this very stringent need. But of course, in other situations, I mean, in my fish, how hungry is it? And how stringent is it for it to find the prey? That depends on the animal, on its history, and that's something that we've started to do. We've planned for it for a system that didn't really work, but try to find out how the decision process depends on hunger in different systems. We've done it for jellyfish, but it didn't really work because the experimental details just didn't really work. So we're thinking of doing this for higher organisms. My collaborators really like piranhas, for a fascinating system, and we might be working on that. We are looking for the system to do this.
Thank you.
Yeah, sorry, it was not the yes or no answer.
It wasn't a yes or no question. That was great, thank you.
Simon, yeah, do you want to?
Should we shift to the, I guess we'll just